Hello students, this is Dr. Faizan Mirza. I am discussing with you some working definitions of genetic technology. What is genetic engineering? Genetic engineering is a procedure in which the genetic information of an organism is changed by altering the base sequence of a gene or introducing gene from another organism into this organism or this cell. Genetically modified organisms or GMO are organisms whose DNA has been changed in a way that does not occur naturally or by selective breeding. We actually play with genes, we change the genes, we edit the genes, we just add or remove the genes. Vectors are agents that you are used to deliver the gene that is to be added into a cell. For example, plasmids, which are in bacteria, virus, which can the gene can be added in the nucleic acid of the virus. Liposomes, these are artificial vesicles of phospholipid, and they can carry the gene as well. Restriction and their nuclease, for example, EcoR1, BAMH1, Cas9, these are all used to cut nick DNA molecule at a specific site called a restriction site. Recombinant DNA, rDNA, the DNA produced by adding or editing gene to existing DNA, is basically the recombinant DNA. It is artificially produced by adding DNA from two different species or two different sources. Transgenic organism. Transgenic organism is an organism with a gene added to its cells that initially did not have that particular gene. What's a gene probe? It's a length of a single-stranded DNA sequence having sequence complementary to another piece of a single-stranded DNA, which is being detected. Hybridization is the binding of two single strands of complementary bases together. This brings us to the concept of microarray, gene chip, DNA chip. Microarray are slides with thousands of tiny positions with each spot containing a known single strand of DNA fragment gene. The DNA fragment acts as probes that can detect complementary sequences of DNA or RNA. Gel electrophoresis. Gel electrophoresis is a technique in which we separate the charged molecules, DNA and proteins, through gel in an electric field. The distance covered by the molecule being separated is dependent on the mass of the DNA fragment or RNA, the mass of that fragment. The distance covered by molecule being separated is dependent on not only mass but on charge as well, but in which case when we are using proteins, because proteins have different R groups and they add to the charge and the mass as well. PCR, polymerase chain reaction. Polymerase chain reaction basically is a technique to amplify DNA sequences in very large quantity, and that's PCR. But PCR, one needs to keep in mind that when we are using PCR, we use Taq polymerase. Taq polymerase, there is no proofreading involved. So Taq polymerase will keep on just copying a DNA into several copies. If a mutation takes place, if the copying is incorrect in some way, a huge quantity of abnormal DNA will be copied again and again because there is no proofreading mechanism. In our cells, when the DNA is replicated, we have different DNA polymerases which actually work by proofreading as well. So PCR, although very useful, but mutations can accumulate in PCR. The concept of promoter. What's a promoter? Promoter is a length of DNA molecule where RNA polymerase binds during transcription. We know transcription is how when the DNA template is transcribed into mRNA. Gene expression can occur only when RNA polymerase binds to the promoter region in the prokaryotes. And in case of a eukaryote, the RNA polymerase together with the transcription factors must bind to the promoter region. So that's the difference in the eukaryotes and the prokaryotes. What's gene expression? Gene expression deals with the transcription of the template strand into a polypeptide. We know that the gene gets expressed, the transcription of the template strand into mRNA, not the polypeptide, into mRNA, because this is gene expression. Once the mRNA is being transcribed, then the mRNA will go from the nucleus to the ribosomes and it will be translated into a polypeptide, because proteins are made after gene expression. So gene expression actually accounts for turning the DNA template strand into an mRNA. There's transcription followed by translation, in which the template mRNA goes to the ribosome, where translation proceeds and the polypeptide is made. And the polypeptide adds a trait to the cell. What is gene editing? Gene editing is a form of genetic engineering. And this form of genetic engineering, in this the genome of the organism of the cell, it can be altered by either deleting, inserting, or replacing the gene or allele present in that cell or in that organism. That basically is gene editing. We use CRISPR-Cas9 system for this, and the CRISPR-Cas9 system is actually coming from bacteria. And why bacteria have CRISPR-Cas9 system? Because bacteria are often coming across viral infections, and the viral infections in this case are from bacteriophage virus. This virus infects bacteria. Bacteria have CRISPR and Cas9. The CRISPR-Cas9 can cut the viral DNA, and it can survive the viral attack. So we use the CRISPR and Cas9 system for our own benefits in gene editing. There's a small concept of eugenics that you just need to recall. Eugenics is just encouraging some people to have children and to discourage others from not having children, and this because we know that we actually are aiming to get a much improved human population. For example, individuals having Huntington, they can be asked not to have children because they may pass on the Huntington allele to the next generation, and even single dominant allele of Huntington can cause Huntington disease. That's eugenics, because we want a much healthier population to continue. Although eugenics, there are a lot of ethical and social and moral issues regarding it. So although the concept is there, so whether it should be used or not used, there's a huge debate about it. Bacteriophage. What are bacteriophage? Bacteriophage viruses are viruses that can infect bacteria, and when these viruses infect bacteria, they have a double-stranded DNA, and phages, they insert this DNA into the bacteria. The double-stranded DNA then gets incorporated into the genome of the bacteria, and this just becomes a part of the bacteria then. And this is exactly the double-stranded DNA of the virus of the phage that CRISPR-Cas9 in the bacteria can cut, so it can survive the viral attack. What is gene therapy? Gene therapy is treatment of a genetic disorder by inserting genetically corrected cells into body or by introducing functioning genes directly into the cell. So that pretty much sums up all the working definitions for this particular chapter. Thank you so much.